Welcome back to Journal Club, and today we're talking about Nidovirus in snakes. And this is one of those special post-conference Journal Clubs where I talk about something cool written by someone I met. So snakes, my favorite group of critters, are reptiles in the order Serpentes. They are squamates, which technically makes them legless lizards, except that legless lizards are something completely different that kind of look like snakes but aren't. Isn't taxonomy fun? Turns out lots of things have adopted that long, skinny body plan because it works. But snakes are special because they have a bunch of cool things that set them apart, like their skull has more joints compared to a normal lizard, allowing them to swallow much larger prey. And then also their paired organs appear one in front of the other rather than side by side, which helps them be long and skinny and snake-like. Often imitated, never replicated. I guess to summarize, snake, different snake, best snake, not a snake, not even a lizard. Just get out, you little rascal. Naturally, like any other species on the planet, snakes have their own viruses, and one of the most significant groups is the Nidovirales. This order of viruses has been reported in mammals, birds, fish, crustaceans, and insects, so a whole bunch of different things, and so it makes sense that they'd affect reptiles too. I mean, also, birds are technically reptiles, which I think means my grandma, out feeding the ducks was actually an avid field herper. So, basically this. Ignoring all the angry birders in the room, a snake-associated Nidovirus, now known as Serpentivirus, was discovered in 2014 and linked to respiratory disease in pythons. And so the whole goal of this study is to take that virus and confirm that it actually causes the disease that it's linked to, which is important because there's a ton of organisms out there that may just be bystanders in disease. If you recall when we talked about Robert Koch and tuberculosis, Koch's postulates are a foundational concept in microbiology. Briefly, he says that the culprit should be found in all organisms suffering from the disease, it should be isolated from the diseased organism, cultured, and then the cultured microorganism should induce the same disease when reintroduced into a healthy specimen, and then it should be re-isolated. There's a whole bunch of exceptions, so the rules have been bent and adapted as needed, but it is still a foundational concept in proving that an organism causes a disease. Now, in order to grow Nidovirus in the lab, they had to have an appropriate cell culture. Viruses are a bit of a pain. If you're tracking my words of wisdom, that one's probably right up there with heart disease is bad for you. And Gen Z, if you adapt my words of wisdom into some sort of Tuesdays with Maury, I want Danny DeVito to play me in the film adaptation. Anyway, what makes viruses especially annoying is that they won't grow in a normal culture plate like a bacterium. That's why we had such a hard time discovering them in the first place. And so you need to culture appropriate host cells to grow them if you want to fulfill that first part of Koch's postulates, which is what they did. So they had a diamond python that had spinal disease and was euthanized, and they were given small sections of the myocardium, which they then used to generate a cell culture. They then inoculated the cell culture with Serpentivirus from the ball python colony where it had been first identified in 2014. And then with the cultured virus, they then inoculated 3 ball pythons. And then finally, they used the same culture protocol to re-isolate the Serpentivirus from the experimental snakes. And so within a couple weeks, the infected snakes developed red oral mucosa and excessive mucus. Over time, they developed more severe oral swelling and petechiation and increased respiratory effort. So a stomatitis, and specifically there is inflammation around the glottis, and a respiratory infection. And histologically, this corresponds to significant inflammation in the oral and nasal mucosa. So here the oral mucosa is attenuated to ulcerated and just jam-packed with inflammatory cells, mostly heterophils with fewer lymphocytes and plasma cells. Likewise, the mucosa of that trachea and esophagus is also eroded in places and packed with similar inflammatory infiltrate, but there's also this proliferative response in the esophageal and tracheal mucosa. And the lung has what's called an interstitial pneumonia, or inflammatory cells within the walls of the faveoli, which are basically the reptile version of the sacs that help them breathe, or alveoli that we'd have in mammals. And likewise, there's also proliferation of the pneumocytes, which is probably analogous to type 2 pneumocyte hyperplasia in a mammal. And in order to prove that this is caused by the virus, they also did immunohistochemical staining for the nucleocapsid proteins. So that's what the red signal in these images is. And you can see that there's red in the oral mucosa, the trachea, the esophagus, and the lung, versus tissue from a normal snake on the bottom has no signal in it. Interestingly, they also found it in the stomach and intestines, so that may be a route of transmission for these snakes. So this shows that inoculation of that isolated Serpentivirus causes disease when they introduce it to a new ball python, proving Koch's postulates for Serpentivirus. And specifically, they noted stomatitis, tracheitis, and interstitial pneumonia as key lesions. Previous reports from the naturally infected colony had a greater degree of pneumonia, likely associated with it being a more chronic infection. These snakes have a surprising amount of respiratory reserve capacity, and so chronic pneumonia can brew for a really long time, and the snakes compensate by spending more time on the cold side, which limits their oxygen consumption. They also suggested that differences in husbandry between the laboratory condition, which followed published best practices, and the breeder condition may have contributed. So, for example, differences in temperature and humidity between the two setups may have altered susceptibility to secondary infections, or depending on how the enclosures are set up, if the snake can't move around to change temperatures, well, that may change that behavioral compensation for the pneumonia.